is that and truth being said, I'm a pharmacokineticist, so a bit tunnel visioned. Is there a role for studying intracellular kinetics of small molecules? If I look at these diseases, you obviously optimizing the drugs to go into the liver, but within the liver, there's liver cells, different liver cells, and within the cells, there are certain targets. So is that something that we could even account for during the entire drug development process? Do you think so?
So we, what's driving that concentration in the liver? There are drugs like sofosbuvir, which are actually designed to take advantage of a certain mechanism to deliver it and trap the molecule in the cell, right? In HCV. For HBV, we've fine-tuned all the characteristics of the molecule to get this liver centricity. Now, what is driving that liver centricity? Some general characteristic of the molecule or some active uptake of the molecule into the liver, we don't know at this point in time.
Right. I have another question. So the small molecule PD-L1 inhibitor is like very interesting, and is it also very likely that it has activity in other diseases, but also that it might actually cause some side effects in that respect?
Well, we've been very interested in not only the PD-L1 small molecule for HBV, but we have a particular interest in hepatocellular carcinoma as a potential utility for these molecules, right? So again, they're highly liver centric, right? Versus an antibody. So could that be beneficial to cancer patients or HCC patients where they have very few options at this point in time still? So that is a possibility as another therapeutic target area for us. But I think the possibilities are endless really for these small molecules and as checkpoint blockade inhibitors. So
I have one final question. So you showed repetitively ALT elevations at around weeks 3 to 4 with a lot of different small molecules that had an effect in hepatitis B virus infection. Did you investigate what it actually is that causes that? Or because it almost seems like it might be an effect that you see in every patient, and it may be a sign of the activity of the drug itself.
That's a good question. We really wanted the answer to that because we thought we had the answer when we looked at the second generation molecule 506, and what that molecule we saw, even though it was a small metabolite formed, that we saw in the Ross ATP assay that was pretty active. So we thought it was a metabolite. We believed that we had an assay to address that, to screen for that, and we put our next generation agent through that, and it was absolutely clean, and metabolites were absolutely clean. So we were then really surprised in the end when we saw this, especially when we had long-term toxicology studies that showed there were no liver effects at all with this the third generation agent. We don't know. We just don't know what it is, and that's we would love to figure that out, but at this point in time, it's going on to other things.
Okay, thank you. If there are no further questions, then I'd like to thank you very much for a very inspiring talk. Thank you.
Good afternoon, everybody. I'd like to welcome you to this oral communication session on cardiovascular. My name is Cherry Wainwright. I'm from Robert Gordon University in Aberdeen, and my pleasure to be Vice President for Meetings for the British Pharmacological Society. And this is my co-chair.
Thank you for the opportunity. I'm Monica Mukhia and recent graduate from University of Glasgow, Masters in Clinical Pharmacology. Thank you so much.
Okay, thank you. So our first speaker this afternoon is Paraskevas Zampas from the Biomedical Research Foundation of the Academy of Athens, who is going to speak to us on hydrogen sulfide donors' improved cardiometabolic profile in a mouse model of metabolic syndrome.
Thank you very much. I'm actually substituting for my student who couldn't make it to Glasgow. I'm the senior author, I'm Andreas Papapetropoulos, and I'll be talking to you about hydrogen sulfide donors in cardiometabolic syndrome. The metabolic syndrome is a clustering of medical conditions that are associated with problems in the cardiovascular system or in metabolism. So to be classified, for humans to be classified as having metabolic syndrome, they need to have a blood pressure of over 130, or fasting blood sugar over 100, a waist larger than 40 inches, and abnormal values for HDL, abnormally low values for HDL, and abnormally high levels of triglycerides. There's a growing number of people throughout the world with metabolic syndrome, and to introduce our molecule of interest and our pathway of interest, which is hydrogen sulfide. The hydrogen sulfide pathway consists of a number of enzymatic reactions that will give rise to hydrogen sulfide, and a number which are depicted on the left-hand side, and a number of enzymes and pathways that will degrade hydrogen sulfide. On the left-hand side, you can see enzymes that the transsulfuration pathway, CSE and CBS. What is my pointer? This is my pointer, right? Okay. The CSE and CBS that are part of the transsulfuration pathway using homocysteine or cysteine to generate hydrogen sulfide. There's a third enzyme in the pathway, 3-mercaptopyruvate sulfotransferase, that uses a breakdown product of cysteine, 3-mercaptopyruvate, to yield hydrogen sulfide. There's additional sources of hydrogen sulfide in our bodies. H2S can be made through gut microbiota and also through non-enzymatic reactions, and there's also the H2S that we consume. On the right-hand side, you can see the pathways that are responsible for breaking down hydrogen sulfide, and the main enzymes here are sulfide quinone oxyreductase, abbreviated as SQR, ethyl-1 rhodanase, also known as TST1, and sulfide oxidase. So hydrogen sulfide has been linked to a number of conditions that are associated with the metabolic syndrome: obesity, hypertension, type 2 diabetes. Hydrogen sulfide is usually linked in a therapeutic way with these pathways, meaning if you provide hydrogen sulfide, you are able to ameliorate obesity, reduce hypertension, and improve the complications of type 2 diabetes. Our own group has done quite a bit of work on hydrogen sulfide and obesity. We find reduced levels of hydrogen sulfide in obesity, and especially the enzyme MPST, or 3-mercaptopyruvate sulfotransferase, is responsible for restricting excessive weight gain in animals fed a high-fat diet. So with this in mind, we asked two questions. We asked whether endogenous levels of H2S are altered in the metabolic syndrome in a mouse model, and whether we can, if they're altered, whether we can correct the parameters of metabolic syndrome using pharmacological approaches. A few words about our model. We use a mouse model where we feed mice a high-fat diet, 40% of calories from fat, for 10 weeks. Then we introduce LNAME to inhibit endothelial nitric oxide synthase for an additional 5 weeks, and then look at the end of the 15 weeks what happens to their body weight, blood pressure, and hyperglycemia. This looks very much like the two-heat model that is used in the HFpEF literature described by Joe Hill's group. Only these mice in the heart failure model are fed a much higher high-fat diet with 60% of the calories coming from fat and L-name throughout the duration. This model that I'm just describing does not lead to the development of heart failure with preserved ejection fraction. So a characterization of our model in this first slide: mice receiving a controlled diet don't gain as much weight. The mice that receive a high-fat diet gain more weight. L-name doesn't really change that. And in terms of blood glucose levels, you see that obesity leads to impaired glucose handling and increased and glucose tolerance. And this is obvious in the mice with a metabolic syndrome. Obviously, due to the L-name administration, we also have increases in systolic and diastolic blood pressure in the groups of mice receiving L-name. And surprisingly, only the group that has metabolic syndrome has an increase in the heart weight to tibial length, suggesting cardiac hypertrophy, and increased in mass in the different adipose tissue depots, inguinal and gonadal white adipose tissue. So we do have a model that's working, and it recapitulates the features of metabolic syndrome in humans, and we went ahead and characterized the levels of hydrogen sulfide and the different enzymes in the aorta and in the heart. So looking at the H2S synthesis enzymes, we observed that CBS and MPST are reduced in the aorta of mice with metabolic syndrome, and also the enzyme that's responsible for breakdown, TST, is also reduced in these mice. Unlike what we observed in the aorta, the levels of the enzymes for both the synthesis and the degradation of H2S were not altered in the heart. Given that we found reductions in both H2S synthesizing enzymes and H2S degrading enzymes in the aorta, we went ahead and measured actually H2S in the tissues, and there's two different pools. There's free sulfide and there's sulfane sulfur. Both of these pools were reduced in mice with metabolic syndrome, in line with our observations that no changes in the enzyme levels of H2S synthesizing or degrading enzymes occurs in the heart. We observed no change in the levels of H2S in the heart. So we have established that the levels of H2S in metabolic syndrome go down in this mouse model, and we wanted to see if administering H2S donors would correct the parameters of the metabolic syndrome. So in this slide, we introduced the treatments. We do the 10 weeks of high-fat diet, and then on week 10, we introduce L-name to increase blood pressure, and at the same time, we give either GYY 4137 in IP or SG1002 in the food. Both of them are hydrogen sulfide donors, and GYY is a hydrogen sulfide donor, SG is more of a polysulfide donor. What we observed is that the mice receiving the sulfide donors have reduced body weight, and they have somewhat improved glucose handling as judged by the glucose tolerance test. We have also observed that the mice receiving GYY have reduced systolic and diastolic pressure. There's a trend for the SG group to decline, but this did not reach significance. So also in the heart, the hydrogen sulfide donors, especially GYY, can reduce the heart to weight, and this treatment with the sulfide donors did not alter the mass of the adipose tissue depots that we studied, inguinal fat and gonadal fat. In the heart, GYY was able to reduce cardiac fibrosis, as depicted on this slide, during metabolic syndrome, and the increase in blood pressure. This leads to fibrosis in the heart, and we were able to reverse that with GYY. There's also, again, a trend here with the SG compound, but this does not reach significance. To conclude, metabolic syndrome causes changes in hydrogen sulfide metabolism, resulting in a reduction of endogenous H2S levels in the vasculature, and pharmacological interventions based on H2S restoration could be effective in metabolic syndrome treatment. I'd like to end by thanking our collaborators in this study, the funding sources, and this is a very important slide for me. We'd like to invite you to the EFAR conference 2024 in Athens next year. Thank you for your attention.
I see somebody is already at microphone number 2. If you would like to ask a question, if you could make your way to one of the standing mics, and I will keep an eye open. So, yes.
Rebecca Ritchie, Monash. Thank you for a nice, clear presentation. Have you looked at the effects of H2S metabolism in smaller vessels, the coronary, microvasculature, resistance vessels?
We'd love to, but we have not. I know that the relevance would be in the smaller vessels, and some of our ongoing studies to study contractility and how that is impaired and endothelial dysfunction are unfortunately in the aorta. We'd love to collaborate, though.
Thanks. And a very quick follow-up question. Have you looked at perivascular fibrosis?
We are looking at that, but these studies have not been completed. That's a very good idea. Thank you.
Okay, one here at 2 and then at 3.
Thank you. I'm from Mexico City, and I wonder if you have tried to look for reactive species, oxygen species, for this effect of hydrogen sulfide.
This makes perfect sense. Your suggestion is in line with what we would expect in metabolic syndrome, increased levels. We have not measured them. We would expect they are increased, and we would expect that supplementation with donors, hydrogen sulfide donors, would actually ameliorate that, but we have not measured it. We've measured it in other contexts, in different types of experiments, in obesity or in heart failure, but we've not done it in this particular model.
Yes, thank you.
Thank you very much, Andreas, for a nice presentation. This compound, this H2S donor, by itself, does it change blood pressure or heart rate?
The short answer is it depends on which of the two you are, SG or the GYY?
Yes, both.
Okay, but the GYY will reduce blood pressure, and SG will cause a decline that in our model is not statistically significant. Throughout the literature, at this dose, which is probably the maximum dose that has been used, doesn't do too much in the blood pressure. To get a reduction in blood pressure, you need heroic concentrations, and the entire field with H2S and hydrogen sulfide, as you probably know, in terms of regulation of blood pressure, has lower concentrations doing one thing and higher concentrations doing another.
Can I just ask a question? I think, did you say at the beginning that this model, you don't see the functional changes that you would expect with HFpEF?
Yes, that's correct.
But later on, you showed that there's quite clear hypertrophy and cardiac fibrosis, which I would have imagined would result in some cardiac dysfunction.
Thanks for pointing this out. When we do the echo measurements and we look at ejection fraction, there is no difference. And when we do the E to A prime and E to A ratios, this is not different either. There is this hypertrophy. So you don't have the classic, we haven't measured the pressures.
So you've not done like pressure volume loop or anything?
Yeah, we haven't done all of this, okay. but echocardiographically, it seems that this model does not drive the animals into heart failure. Now, if you're taking away enzymes like you use 3-MST knockout mice, you'll drive them into heart failure. So I would say it's a borderline, the best I can describe.
Okay. Thank you very much.
Thank you.
Thank you so much. Going onward to our session, we would like to welcome Mohammad Adim Abdul Ghani on carboxyl amylorides, the cardiac fibrosis and the hypertrophy in doxorubicin-induced cardiotoxicity rat model.
I'm just getting a signal that I don't think that speaker is here. Is that correct? Yeah. Okay. I'm just conscious of the fact that we are supposed to be staying on time, parallel with the other sessions, so people can move backwards and forwards. So I think my best option is to take a short break to allow people to move between sessions so that we keep to time. So apologies for this, but we will reconvene in just over 10 minutes. Thank you. Apologies, there seems to have been some confusion or the slides that the speaker put in did not appear to have been uploaded properly. So what I'm going to do is I'm going to push that communication back to the end of the session. I know it will go into the coffee break, but at least we'll keep on time for the other presentations. So please bear with us and apologies. As they say, the best laid plans of mice and men, which is a good phrase, I suppose, for a pharmacology meeting. There apparently had been a last-minute change of a speaker presenting the previous abstract, so we are hoping to have that included at the end of this session. We know it will run into the coffee break, but at least we can make sure that that person gets the opportunity to present. So I'm going to hand over to my co-chair now to introduce the next speaker.
So going forward, let's welcome Baofeng Yang from Harbin Medical University on topic on new targets for cardiac ischemia for fusion injury, LncRNA. Let's welcome Baofeng Yang.
Okay, thank you.
She will be here in a couple of minutes.
No, I'm sorry. We need to stick to the time for the actual speaker, so we need the speaker here now. Thank you. Do we have our speaker yet? Okay, I'm sorry, but I think we're going to just have to move on to the next speaker, and we'll wait, see if this other one turns up. So apologies for the mistiming then, but the next speaker is, who I hope is here, is José Britto-júnior from the State University of Campinas to speak on 6-nitrodopamine is the most potent endogenous positive inotropic agent in the rat-isolated heart. Thank you very much.
First of all, I would like to thank organizers the opportunity to present the work you have been developing on 6-nit dopamine. For the last couple years, you have been investigating the release and action of 6-nit dopamine in various organs. For instance, 6-nit dopamine is released by human umbilical arteries and they are detected by liquid chromatograph compared to tender mass experimentally, LC-mass. In the human umbilical cord vessels, 6-nit dopamine acts as a potent vasodilator, antagonizes the contracting response of dopamine at the Q-like receptors. In head-isolated right atria, present basal release of 6-nit dopamine, whereas 6-nit dopamine acts as a potent positive inotropic agent in these some cases. Actually, 6-nit dopamine is 100 more times potent than noradrenaline and adrenaline as a positive chronotropic effect, and 10,000 more potent than dopamine as a positive chronotropic agent. Another interesting characteristic of 6-nit dopamine in the head isolated right atria is that 6-nit dopamine presents a remarkable ability to synergize with noradrenaline, adrenaline, and dopamine in the positive chronotropic effect induced by the classical catecholamines. As measured by LC-mass, 6-nit dopamine is released by rat isolated ventricles, and as described before, the release of 6-nit dopamine is reduced when the ventricles pre-incubate with the nitric oxide synthase inhibitor, LNM, and when the ventricles were obtained from animal chronically treated with LNM. It is important to indicate that incubation with LNM did not a significant reduction in the basal release. 6-nit dopamine, basal release 6-nit dopamine, but it did not abolish, indicate that the other biosynthetic pathway for 6-nit dopamine. Interesting, I showed this slide, incubation the ventricle with the sodium channel blocker, tetrodotoxin, did not affect the basal release of 6-nit dopamine, indicate that the release of 6-nit dopamine is unlikely to be from neurogenic source. Rat isolated to heart Langendorf, heparin as injected to prevent blood control, and anesthesia was performed by isoflurane overdose. The isolated heart was performed with chloride solution, equilibrated with carbogen gas mixture, and constant flow 10 ml per minute, and the pressure 80 to 10. As to recollect the Langendorf preparation, here is a short illustration of the said procedure. This is a typical trace obtained from the isolated hearts. Here, it can be observed the increase in left ventricular developed pressure induced by a single bolus of 6-nit dopamine at the dose 1 picomole, and the increase in left ventricular developed pressure induced by a single bolus of noradrenaline, adrenaline, and dopamine at a dose of 1 nanomole. Yes, 6-nit dopamine is 1,000 times more effective than other classical catecholamines. Here, using the head perfusion heart model, a single bolus eject of 10 femtomole of 6-nit dopamine causes a significant increase in the left ventricular developed pressure. At this dose, 6-nit dopamine is 1,000 times more potent than the classical catecholamines, noradrenaline and adrenaline, and 10,000 times more potent than dopamine as a positive inotropic agent. Again, observing with the inotropic effect 6-nit dopamine is the head isolate heart rate. The effect is apparently not dose dependent. Here are the results obtained with the maximum rate of result of the left ventricular developed pressure, the dp/dt max, on more the left ventricular developed pressure itself, a single bolus inject of 10 femtomoles of 6-nit dopamine causes a significant increase in the dp/dt max. Furthermore, here are the results with the rate pressure product, defined as the product of heart rate and left ventricular developed pressure, as seen before with the left ventricular developed pressure itself. A single bolus inject of 10 femtomole of 6-nit dopamine causes a significant increase in the pressure product. As noted the introduced slide, 6-nit dopamine has a potent inotropic positive action in the head isolate atria, and it has a remarkable ability to synergize with noradrenaline, adrenaline, and dopamine. For this slide, we have infusion for 1 minute 6-nit dopamine at the final concentration of 0.1 femtomole. At this concentration, 6-nit dopamine does not present positive inotropic action in the head isolated heart. However, a single bolus of 0.1 picomole of noradrenaline causes substantial increase in the left ventricular developed pressure. As expected in the slide, the minimum dose of noradrenaline to increase left ventricular developed pressure of 10 picomole. This remarkable potent of the inotropic effect of noradrenaline induced by 6-nit dopamine, as was observed with adrenaline and dopamine. In conclusion, the result demonstrates that 6-nit dopamine, besides being the most potent endogenous positive inotropic agent so far described, has a remarkable ability to synergize with the classical catecholamines. 6-nit dopamine presents a novel therapeutic approach for the treat whether acute or chronic heart failures.
Thank you very much. Do we have any questions from the audience? Yes, mic 3. Microphone 3.
Ulf Simonsen from Denmark. I was just wondering whether you can block the effect with a beta blocker.
the beta block.
Yeah.
The beta block abolish the increase the inotropic action of sedative dopamine, but is more important than this dopamine, either adrenaline and the dopamine. For example, atenolol 10 nanomole don't block adrenaline, noradrenaline, and dopamine action inotropic in the Langendorf, but abolish the effect of 6-nit dopamine.
I'm not sure I understand you, but if you take propranolol, for instance, and infuse that in your preparation, can you then block this 6-nitrodopamine response?
Propranolol, I don't use propranolol here, but so atenolol.
Okay, fine, thank you.
Thank you.
We have in mic 2, yes.
Rebecca Ritchie, Monash University. What can you tell us about the vehicle you used, and did that impact pH of the buffer? And what are the pharmacokinetics of 6-nitrodopamine?
Me.
Okay. What did you dissolve 6-nitrodopamine in?
What is desserts?
What did you make it up in?
Sedative dopamine is released from umbilical.
Yeah, when you were adding it to, you said you added the 6-nitrodopamine.
Water. It's diluted in water.
Thank you, thank you, thank you.
Water.
Thank you. I have a challenging accent. If you're proposing to use 6-nitrodopamine for chronic heart failure, what are its pharmacokinetics in vivo? How long does it last? Is it broken down? Is it cleared?
Well, we haven't been able to measure 6-nitrodopamine in blood levels so far because it's so potent, so the concentration must be very, very low, okay? So we haven't been able to detect circulating levels of nitrodopamine. So at the moment, we have commissioned to make a label nitrodopamine in order to be able to study the kinetic of 6-nitrodopamine half-life in vivo. However, the effect is very long-lasting.
Thank you.
Thank you.
Okay, thank you very much. I think a little bit of confusion has been cleared up, and in fact we do just have one more speaker this afternoon. The topic is new targets for cardiac ischemia reperfusion injury. And can I invite Honglin Xu to the stage to give the presentation?
Hello, everyone. So today's speaker should be the Professor Yang, but because some personal reason he can't come. So on behalf of him, I will give you a talk. So today I'm going to talk about the role of LncRNA mediated epigenetic regulation in the heart injury. Cardiovascular disease remains the leading cause of deaths in the world. The numbers of deaths owing to cardiovascular disease is about 80 million in 2020 all over the world, and estimated to be 23.6 million in 2030. Statistic data revealed the prevalence of cardiovascular converts from the rising burden of cardiovascular disease, ischemic heart disease, and ischemic stroke, declining the incidence and the mortality of hemorrhagic stroke. To develop effectively and timely strategy to cope with the challenge of cardiovascular epidemics, since 2010, more than 30 drugs for the treatment of cardiovascular disease have been approved by the FDA. Even so, the identification of novel molecular targets and the development of effective drugs for the treatment of cardiovascular disease remains the current top priorities. 95% of the genome is non-coding RNA, which also indicates the importance of non-coding RNA in the body. Non-coding RNA include structural RNA and regulatory RNAs. Beyond their applications as diagnostic and prognostic biomarkers, non-coding RNA can also be the targets of tools for novel therapeutic strategies. Non-coding RNA have been identified as critical novel regulators of cardiovascular risk factors and cell functions and are emerging as fundamentally novel therapeutics. Researchers have developed complex tools and modulated these non-coding RNA with the aim of establishing novel next-generation strategy to combat cardiovascular disease. Another important non-coding RNA is LncRNA, which also been the focus of research in recent years. LncRNA are a class of non-coding RNA molecules of longer than 200 nucleotides present in the nucleus of cytoplasm. More than 40,000 LncRNAs have been found in human tissues, and according to their position in the genome, LncRNA can be categorized as sense, antisense, bidirectional, intronic, and intergenic. Unlike protein-coding genes and microRNAs, LncRNA are often poorly conserved between species and are highly tissue-specific. LncRNA research started later than microRNA research, and after more in-depth research, LncRNA has also the potential to become a drug therapeutic target. The growing evidence had demonstrated that the LncRNA ZFAS1 and antisense LncRNA to the 5' of the protein-coding gene ZNFX1 had significant role in the regulation of tumor. In our previous study, we identified ZFAS1 as an independent predictor of acute MI, and additionally, we found that ZFAS1 bound to and inhibited the intracellular level and activity of sarcoplasmic reticulum, calcium ATPase 2A, and contributed to the impairment of cardiac contractile function in MI. And last year, we published the paper that LncRNA ZFAS1 promote myocardial ischemic reperfusion injury through DNA methylation-mediated Notch1 downregulation. Therefore, ZFAS1 might be considered as a new therapeutic target for preserving cardiac function. The calcium homeostasis, a critical determinant of cardiac contractile function, is critically regulated by SERCA2A. Our study found that ZFAS1 was rapidly increased in both ISR and the cytoplasm of MI heart, and such an upregulation produced a significant negative effect in MI heart. And at the molecular level, ZFAS1 was found to directly bind to SERCA2A protein on one hand and to repress expression on the other hand. Moreover, the NF-ATC2 account at least partially for the upregulation of ZFAS1 in MI. And our papers have several citations in Circulation Research and Cell Calcium. The myocardial ischemia reperfusion injury has been correlated with oxidative stress, autophagy, ER stress, and epigenetic alterations. Although there are many treatment strategies, the incidence remains high, which indicates it's very important to find new targets. So next is the experimental timeline of the MIRI model. First, the left anterior descending coronary artery was ligated for 30 minutes to cause myocardial ischemic. Then the line was released for 48 hours to allow for reperfusion. And this is the result part. From the left panel, the echo result, we can see in the MIRI mice, the cardiac function was reduced, and ZFAS1 expression was dramatically elevated. And in cardiomyocyte exposed to hypoxia reoxygenation, the cell viability was reduced, and ZFAS1 expression was elevated. And next, we inhibited the ZFAS1 with AAV vectors to examine the ZFAS1 function. So we could see the ZFAS1 knockdown significantly improved miRNA-induced impairment of cardiac function. And furthermore, SHZFAS1 reduced the effect size of miRNA heart. And next, in vitro, we transfected SIZFAS1, and we could see the SIZFAS1 could mitigate the hypoxia reoxygenation-induced reduction of cell viability and cytotoxicity, showed the similar result. The next is the tunnel staining result, which indicate that the apoptosis was restored by SIZFAS1, and the enhanced ROS generation induced by hypoxia reoxygenation was also reduced by SIZFAS1. Next, to understand the molecular processes supporting miRNA by ZFAS1, we constructed cardiac-specific ZFAS1 knock-in TG mice, and we can see that ZFAS1 was upregulated. Then we compared the transcriptomes between TG and the wild-type heart by gene expression profiles. The experiment revealed that 30 downregulated genes and 90 upregulated genes in TG mice. And GO functional analysis also of these 49 differentiate expressed genes was then conducted with the cluster profile bioinformatic tools. So next, we predicted and screened the direct target of ZFAS1 from 4 pathways, including activation of cardiovascular physiology, stress response, programmed cell death, and cardiac development and apoptosis process. And we identified that the Notch1 and the NTRK3 might be the potential targets of ZFAS1. Because Notch signaling pathway is one of the top 30 rich KGG pathway according to a KGG pathway enrichment analysis of differentially expressed. Therefore, our follow-up research mainly discussed the role of Notch1 in miRNA. The Notch1 expression was identified in TG mice to examine the influence of ZFAS1 on Notch1, and Notch1 mRNA levels were significantly lower in TG mice, which is negatively connected to ZFAS1 expression. And similarly, the Notch intracellular domain in TG mice was considerably lower than in the wild-type mice, which was verified by the immunofluorescence. The Notch signaling pathway is involved in a variety of heart functions and is composed of 4 Notch proteins and 5 ligands. The Notch protein is cleaved into the Notch intracellular domain and released into cytoplasm when the ligand on the neighboring cell surface interacts with the Notch receptor. And after being cleaved, the Notch intracellular domain enters the nucleus and binds with the DNA binding protein, which triggers downstream gene transcription, such as Cas1 and HEY. Inhibiting the Notch signaling has been shown to alter the gene supply of cardiomyocytes, leading to cardiac dysfunction. And from the result, we could see in TG mice, the mRNA and protein level of Cas1 and HEY1 downstream regulators of Notch signaling pathway was both reduced and compared with the wild-type mice. And next, we characterized the regulatory effect of ZFAS1 on the Notch signaling pathway in miRNA and found that the mRNA level of Notch1 was reduced during hypoxia reoxygenation, which was reversed by ZFAS1 transfection. And this result showed that inhibiting the expression of ZFAS1 remarkably increased the NICD protein levels. And also in vitro, we used neonatal mouse cardiac myocytes, which exposed to hypoxia reoxygenation damage, and similar expression modifications of HEY1 and HES1 were consistently found among the constructs. So how does LncRNA ZFAS1 regulate Notch1? NOTCH1 is because ZFAS1 is localized within the nucleus of cardiomyocytes. These facts promote us to conjecture that ZFAS1 might regulate NOTCH1 transcription through an epigenetic mechanism, such as DNA methylation. We found a substantial CpG element in the promoter region of NOTCH1 using the UCSC genome browser and mass primer to predict the probable domain of NOTCH1 for DNA methylation. And the methylation inhibitor dramatically enhanced the transcription level of NOTCH1 in cardiomyocytes. And next, in TG mice, the bisulfite sequencing revealed a large increase in methylation at the NOTCH1 promoter region. So how does LncRNA ZFAS1 promote DNA methylation of NOTCH1? LncRNA can limitate the gene transcription by binding to DNA and build DNA RNA triple, which can accrue DNA methyltransferase and increase DNA methylation modification in target areas. So we use the RNA tools to see its expression. We can also see the direct physical interaction between ZFAS1 and NOTCH1 promoter region was also verified by means of chereptide with ZFAS1 antisense as a negative control. And the IP result also shows that the DNMT3B bind to NOTCH1 promoter CpG element was considerable, and according to these findings, ZFAS1 bind to NOTCH1 promoter areas and recruit the DNMT3B to enhance NOTCH1 DNA methylation. Then we used the nucleotide replacement, and we engineered a mutation in the ZFAS1 sequence to disrupt the NOTCH1 binding site and investigate the impact of ZFAS1. However, the cell results show that when the cell transfected with mute ZFAS1, the carried plasmid lost its function, and we could see the capacity of mute ZFAS1 to modify the cell viability, cell apoptosis, and ROS levels was likewise lost. These results indicate that the binding of ZFAS1 to the NOTCH1 promoter region is a critical step in its impact on mRNA. And the right part is the drug discovery part, and because of time limitation, if any of you interested in this part, you can see our papers, which published on GACC. So in summary, upregulated ZFAS1 facilitated the DNMT NOTCH1 interaction and promote DNA methylation mediated NOTCH1 downregulation. And according to our current findings, which explains how upregulated ZFAS1 underpins the gene genesis of mRNA, and by interfering this epigenetic process, the NMN could used to treat mRNA as a potential therapeutic therapies. Okay, thank you for your attention. Yeah.
Considering the time, we can go for one quick question.
Yeah, because of the time, I need to catch my train, so I need to leave.
We have time for one question.
Okay.
Okay, I don't see any questions out there. We have actually overrun our time now, so I would just like to thank you and the remaining, all the other speakers this afternoon for their presentations, and again, apologies for the disruption to the program. Thank you very much.
Thank you so much.